the mirror life that could end all biology. The enzyme readout flatlined the instant the soil extract hit the tray, activity to 0, as if the proteins forgot how to be alive. Breakfast yogurt, same result. Antibiotics, useless. In the lab, nobody spoke. The air smelled faintly of bleach and panic. If these molecules were the wrong hand, mirror sugars, mirror amino acids, our biology couldn't read them. Food turns into gravel. Drugs become decorations. And if this chemistry spreads, hospital routines fail before lunch. One clue had been hiding since the 186s, when a young chemist watched light twist the wrong way through a crystal. To see why, we rewind. What we're actually chasing. We're chasing mirror life. Organisms built from the opposite chirality to ours. D, amino acids and proteins. L, sugars and nucleic acids. The flip toolkit. Louis Pasteur set the stage in the 186s, separating tartrate crystals and finding they bent polarized light in opposite directions. He cracked a deeper rule. Biology picks a hand and sticks to it. Our world chose L for amino acids, D for sugars. Mirror life would choose the other way around, and that single choice turns the locks on every door. Here's the plan. We'll follow the blind alleys and hard lessons to the tests that don't assume our chemistry. Then we'll model what happens if mirror life is real, where it hides, how it meets us, and how we keep everyone fed, treated, and calm. This isn't sci-fi. We'll stay inside biochemistry and geochemistry. You can verify from chirality-aware spectroscopy to mass spec designed for agnostic biosignatures. Think of it as switching off the autopilot we've built into every detector. Why now? Because our tools have biases. Culture media feeds our chiral nutrients. PCR reaches only for our DNA backbones. Even metagenomics pipelines throw away nonsense that might just be sensed with a different hand. Meanwhile, extremophiles keep humiliating our expectations. Life grows in arsenic-laced lakes, under acid fogs and inside salt crystals. That doesn't prove mirror life, but it tells us the map is incomplete. NASA's new focus on agnostic biosignatures like an antiomeric excess and circular polarization gives us a net that doesn't care which hand is waving. The stakes are personal. If mirror microbes colonize a wound, your immune system might not see their sugars. Worse, a sudden dump of their fragments could trigger mirror septic shock, pattern recognition gone haywire because the cues are unfamiliar or dangerously similar in the wrong places. Most antibiotics target our handed ribosomes and enzymes. Flip the chirality and many drugs miss. Food chains break if decomposers can't eat what plants make, or if they flip environmental pools faster than we can adapt. We've also learned restraint. Remember GFAG1, the arsenic life claim? It lit headlines, then collapsed under contamination controls and careful isotope work. That saga set our rules. Isotope tracing, rigorous chiral separations, pre-registered protocols, and independent replication. No shortcuts. Open loops to hold on to. Where could mirror life survive next to us without mixing? What would count as proof that convinces skeptics? If we find it, how do we contain it? And could its immortal mirror proteins become tools for medicine and materials without risking the pantry? Now, back to the coin flip. The coin flip, chirality, and the great asymmetry. To understand why a soil extract can switch off an enzyme like a light, we need the rule Pasteur stumbled into while sorting tiny crystals by hand. He noticed some crystals twisted plain, polarized light one way, some the other. Same atoms, same bonds, yet mirror images that wouldn't stack. That's chirality. Hold your hands together. They mirror but don't overlap. Molecules can be like that. Biology made a choice early on. One hand for sugars in our genetic backbones, the other hand for amino acids in our proteins. After that, everything locked into place. In our world, DNA and RNA use these sugars. Proteins are built from L amino acids. Flip those letters and you're in a mirror kitchen where none of our utensils fit. Enzymes are shaped pockets, locks, that welcome a single-handedness of key. Turn the key around and it won't go in, even though it's the same on paper. That's why wrong-handed molecules can sit right next to the correct ones and do nothing but get in the way. Meanwhile, our detectors cheat. We often assume nature will hand us equal mixes, racemic soups, unless something biases the recipe. But life doesn't run on racemic. Homochirality, a single hand across an entire polymer, is how you get long, stable chains that fold, copy, and catalyze. If a stray wrong-handed amino acid sneaks into a growing chain, the protein can misfold or stall. That's not a minor bug. It's the difference between a cell dividing and a cell failing. So where did the bias come from? There are suspects, not a confession. Some minerals, like calcite, offer. Mirror faces where one enantiomer lands and reacts more easily than its twin. Circularly polarized light from star-forming regions can nudge photochemistry, breaking bonds in one hand more efficiently than the other. Autocatalytic cycles can amplify a small tilt into a landslide. Once a reaction favors one hand, it builds catalysts that favor that same hand even more. In lab flasks, chemists have watched tiny excesses snowball into near-pure-handedness when the reaction feeds itself. The key thread is feedback. Once a hand is chosen, everything downstream doubles down. Here's a micro-story from a bench that learned this the hard way. A team was racing a deadline to synthesize a sugar-linked inhibitor. Simple on the whiteboard, brutal in glassware. The NMR looked fine, the mass was perfect, but every enzyme assay was dead. They cleaned glass, remade buffers, accused the freezer. 48 hours later, a junior noticed the catalog code. The supplier shipped the opposite enantiomer of the sugar. Same molecular formula, mirror image. They repeated the coupling with the correct hand, and the enzyme lit up like a bonfire. One flipped chiral center had cost them 2 days and a week of pride. The lesson stuck deeper than the paper. Nature's locks don't forgive. That choice, L for amino acids, D for sugars, rippled into our nutrition. We can't meaningfully eat the mirror pantry. The glucose tastes sweet but slides through us because our transporters and enzymes ignore it. Amino acids are mostly useless as building blocks for our proteins. We do use a few D-amino acids in bacterial cell walls and signaling, which tells you the chemistry is accessible, but those are exceptions riding on an overwhelmingly left-handed infrastructure. Mirror life, if it exists, would look at our fields and forests the way we look at a glass museum. Interesting, not edible. Now bring it back to that flatlined assay. If a world of mirror molecules touched our enzymes, many would clamp shut. The active sites are asymmetric. Wrong-handed sugars won't bind right. Wrong-handed amino acids won't align. It's not that mirror molecules poison us by default. It's that they ghost through our biochemistry. Flip the perspective, and our antibiotics, tuned to L-ribosomal geometries, might sail past mirror ribosomes without a nudge. Even our immune system reads shapes. Pattern recognition receptors grab specific twists of lipids and sugars on bacterial surfaces. A mirrored set could dodge some alarms and, perversely, trigger others by half-matching like a house key that scratches the lock, but never turns. What nobody knew at the time of Pasteur's tweezers is how early that coin toss probably happened. If two origin of life experiments ran side by side, one on a sunlit shoreline, one deep in mineral pores, they could in principle lock opposite hands and diverge forever. Meanwhile, the deep subsurface brines or chimney walls offer places where a mirror lineage might avoid being outcompeted by our chemistry. Our enzymes can't eat theirs can't eat ours, ecological islands built by geometry. Rarely, time itself can blur the border. Amino acids slowly race-mise, left drifting toward right over geological timescales. But that's a clock, not a bridge for living metabolisms. Here's where it gets crazy in a quiet way. If mirror proteins were made entirely from D-amino acids, our proteases wouldn't cut them efficiently. They'd resist many of our usual routes of decay. In a hospital, that's a containment headache. In a materials lab, it's a feature. Stability starts to look like immortality at room temperature. The same structural rule that blocks dinner can preserve a drug from breakdown or keep a catalyst sharp for months. So the great asymmetry isn't trivia. It's the scaffolding under every bite you chew and every pill you swallow. And it leaves open a profound possibility. Another scaffolding could exist right next to us, invisible to our methods, because we designed those methods for the world we inherited. If a second coin landed the other way, we've been walking past it for centuries. The shadow hypothesis, a biosphere we don't see. Paul Davies gave it a name that stuck, a shadow biosphere. Not ghosts, neighbors we've never learned to greet. The idea is simple and rude at the same time. Life might exist right beside us, yet slip past our nets because our nets were tied for one ocean. Our chirality, our genes, our growth habits. We didn't just bias our tools, we baked our worldview into them. Start with the basics. When microbiologists discover microbes, they usually feed them. Plates, broths, flasks. The pantry is stocked with our handed sugars and amino acids. If mirror lineage walked in hungry, it would starve in a room full of food. The conclusion writes itself: nothing grew. Right, nothing we could feed. Culture bias alone leaves most of Earth's microbes uncultured. Flip chirality, and you've built a locked cafeteria. Genetics didn't save us from that trap. It deepened it. PCR, the workhorse of detection, assumes a phosphate sugar backbone with D-ribose geometry and canonical base pairing. Primers are designed from what we already know. Even universal primers are only universal inside our family. If heredity is encoded on a different scaffold, or if mirror nucleic acids kink at different angles, amplification fails cleanly. Screen reads absent. What it really means is, we didn't speak its language. And then there's the computational filter. Metagenomic pipelines toss anything that won't align across known proteins, motifs, or codon usage. That protects us from noise, chimeras, and lab contamination. But it also discards the very surprises we claim to hunt. A chiral opposite would be disguised as junk. Good housekeeping erases the stranger at the door. Field work teases us with edges. Organisms bloom in monolakes arsenic brines, in acid vents, inside salt crystals, and beneath Antarctic ice. Every time we say nothing can live there, something answers with growth curves. That doesn't prove a mirror lineage, but it proves our imagination is smaller than Earth. Meanwhile, NASA and astrobiologists are pivoting. Instead of asking for our signatures, they're mapping agnostic ones, enantiomeric excess patterns that shouldn't appear without selection, circular spectropolarimetry that flags bulk biological homochirality, and mass spectrometry tuned to whole family chemistry rather than a single alphabet. The point isn't to catch mirror life directly, it's to stop missing the outline it throws. So what would count as evidence, not a headline? G5-1 left a scar that still itches. Announced with razzle, unpicked by isotope tracers and clean controls, it taught a harsh rule. Extraordinary claims need boring data. For a shadow biosphere, that means stepwise proof. Show metabolism that prefers mirror substrates in racemic mixes. Separate enantiomers physically and watch consumption patterns over time with labeled isotopes that can't be hand-waved as contamination. If there's heredity, read it without assuming a D-sugar backbone. Use peptide nucleic acids as binders or mass spec proteomics that doesn't lock to L-amino acid databases. And pre-register the protocol so nobody moves the goalposts mid-game. Meanwhile, consider hospitals and farms. If standard enzyme assays flatline in the presence of certain environmental extracts, we shouldn't just shrug and label it inhibitor X. We should ask whether the assay itself is handed and blind. A mirrored pathogen might not trigger classic culture positives. Wounds could look clean while healing stalls, because degradative enzymes can't bite down. Flip to agriculture and soil labs. If decomposition slows for no clear reason, check the chirality of residual amino acids and sugars. A clipping change in an enantiomeric excess across fields would be a red flag worth dropping everything for. There's also a different kind of risk: false positives from abiotic chiral sources. Minerals can sort enantiomers, sunlight can bias photolysis, industrial chemistry spills chiral fingerprints too. The standard has to be biological coupling, link pathways that maintain homochirality against noise. Life leaves coordinated patterns across molecules, not isolated quirks. Look for networks, not trophies. 3 days earlier than any big announcement, you'd want a quiet lab doing the dull work, repeating growth in racemic media, swapping in mirrored controls, flipping antibiotics to their enantiomeric twins, and measuring whether universal inhibitors fail. If our phages and viruses bounce off, if our ribosome-targeting drugs go inert, that's not proof alone, but it piles weight on the scale. One detail will be crucial later: whether mirror proteins resist our proteases in real time, not just theory. If they do, environmental persistence climbs, and containment becomes a planning meeting, not a paragraph. The shadow hypothesis isn't romance, it's humility. We built a magnificent searchlight and aimed it at ourselves. If another biosphere sits in the dark corners, it's not hiding. We're just not looking with the right eyes. And when someone finally flips the switch, the first glow won't be fireworks. It'll be a boring graph that refuses to go away. The false dawn, the arsenic life controversy. While that was happening in field stations and salt flats, a different story lit up the world and burned out just as fast. December 201. NASA teased an astrobiology announcement that would impact the search for life. Cameras, headlines, the whole stage. The claim, a bacterium from Mono Lake, GFOG1, could swap arsenic for phosphorus in its biochemistry, maybe even in DNA. If true, it cracked open the periodic table and cracked our assumptions about life's fixed parts. It sounded like a backdoor to the Shadow Biosphere. Here's why it hit so hard. Phosphorus is a backbone player. DNA, RNA, ATP, membranes. Pull it out, and most intro textbooks collapse. The idea that a microbe could jam arsenate into those jobs broke rules that felt carved in bedrock. And Mono Lake wasn't random. It's hypersaline, alkaline, arsenic-rich, exactly the kind of niche where weird chemistry might thrive. The story wrote itself. Newspapers ran with alien life, and the internet did the rest. In lab rooms without microphones, skepticism started immediately. Chemists pointed out that arsenate esters are notoriously unstable in water. They hydrolyze fast. If DNA had arsenate in its backbone, it should fall apart between coffee breaks. Microbiologists asked dull questions that matter. How pure were the reagents? How low was the phosphate carryover? What did isotope tracers say about real incorporation versus surface contamination? And could the microbe simply be scavenging trace phosphorus with desperate efficiency, the way many cells do under stress? Within a year, independent teams ran the controls that should have come first. They grew GFAG1 in carefully phosphate-limited media, tightened contamination, and layered in radioisotopes and mass spectrometry. The result, the bacterium tolerated high arsenic, but still depended on phosphorus. No arsenic backbones in DNA, no biopolymer swap. The early signals were artifacts, carryover phosphorus, impurities, misinterpreted mass peaks. The study that launched the frenzy got picked apart in comments, replies, and years later, formal retraction. Careers were dented, trust took a hit, and arsenic life became a cautionary hashtag. This wasn't just a pile-on, it was a lesson plan the field needed. Extraordinary biology doesn't get to skate by on unusual growth curves or provocative phrasing. You need isotope tracers that follow atoms into bonds. You need rigorous chiral and chemical separations to rule out lookalikes. You need pre-registered protocols, so post hoc rationalization can't move goalposts. And you need independent replication. Other hands, other benches, same outcome before the press conference. Unknown to them, across town a different group was laying out agnostic life detection frameworks that would have averted some pain. Design assays that don't assume a single backbone, use multiple orthogonal measurements, mass spectrometry to profile families of compounds, NMR to see connectivity, chiral chromatography to separate enantiomers instead of averaging them. Build negative controls that mimic every contamination path you can imagine. Then try to break your own claim. Here's the part that matters for mirror life. The arsenic saga didn't kill curiosity, it forced discipline. If someone brings you a mirror microbe tomorrow, the bar is high and specific. Show growth in racemic media where only one enantiomer disappears, tracked by stable isotopes, so the math isn't hand-wavy. Demonstrate that our proteases don't digest its proteins in real time with controls that prove the proteins are debuilt. Flip our go-to antibiotics to their opposite enantiomers and see if activity swaps as predicted. Use peptide nucleic acids or other achiral binders to fish out heredity without assuming D sugars. Publish the plan before the run. Invite rivals to replicate. There's a human layer too. The NASA rollout magnified a normal scientific tug-of-war into a culture war. Hype promised an answer the data didn't earn, and the correction looked like embarrassment rather than progress. The fix isn't silence, it's pacing. Let the boring graphs pile up. Let the controls get so tight they squeak. Then tell the story with the caveats intact. The audience can handle nuance. What breaks them is the whiplash. In the aftermath, we got better tools and thicker skin. The arsenic lake still shimmers. Extremophiles still surprise us. And the Shadow Biosphere hypothesis survived because it wasn't tied to that claim. If anything, the false dawn clarified what sunrise has to look like. No fireworks, just a stubborn signal that survives every way we try to erase it. Now with guardrails in place, we can build the net that doesn't assume our chemistry and see what it catches. Building the net, how you'd actually find mirror life. With the guardrails bolted on, we stop assuming and start fishing. The trick is to build a net that doesn't prefer a hand. Culture independent first. Mass spectrometry has carried spacecraft and clean rooms for decades. Now it has to carry our blind spot. Instead of scanning for a short list of familiar metabolites, you profile entire families, masses, fragments, and isotopologues without locking to L-amino acid databases. Time of flight or orbit trap systems, coupled to soft ionization, map the chemical zoo. Tandem MS peels the onions. If biology is selecting one hand, you'll see odd depletion patterns in a racemic feed or recurring series that match peptides only if you flip every residue. That's an agnostic hit. Meanwhile, NMR gives you geometry without prejudice. It doesn't care about your alphabet, it cares about how atoms sit and wiggle. Two-dimensional spectra reveal connectivities, coupling constants, and the subtle shifts that betray stereochemistry. Pair that with circular dichroism and vibrational circular dichroism, bulk signals of homochirality, and you're sniffing for the one property we expect life to flaunt, no matter which toolkit it chose: consistent handedness across polymers. A racemic soup cancels itself. A living system leaves a tilt. But graphs don't eat, so you bait. Enzyme assays can be turned into interrogation rooms if you set them up in pairs. Put out L-sugar-specific enzymes beside these sugar-resistant versions and watch activity when you add environmental extracts. If the wrong assay lights up and the right one dies, you've got a clue. More rigorous, build mirrored enzyme pairs where the only difference is the chiral center of the substrate. Track activity over time with stable isotopes, 13C or 2H, embedded in just one enantiomer. Then follow the heavy carbon into downstream products by mass spec. If the heavy label migrates only when the mirror enzyme is present and not when the native one is, you're seeing preference, not inhibition. Now set the table fairly. Racemic media, 50-50 buffets of amino acids and sugars, remove the starvation excuse. Seed parallel microcosms with your sample. In one, the D enantiomers carry a heavy isotope. In the other, the L ones do. If respiration rises while only one label pool disappears, you've separated appetite from artifact. Do it under anoxic and oxic conditions, warm and cold, so you don't confuse thermodynamics with taste. Every time you flip which hand is heavy, the consumption pattern should flip too, where you're looking at chemistry, not life. Physical separation makes the accounting honest. Chiral chromatography columns split twins that look identical to most machines. After incubation, you pass the leftovers through those columns and quantify which hand remains. If biology favored one, the chromatogram shows a clean asymmetry, where a racemic control stays even. Stack the methods: LC-MEMS for quant, NMR for structure, chiral LC for separation, isotope ratio, MS for tracing. Each orthogonal angle closes a loophole GI-AG1 slipped through. Heredity can't hide forever if you don't force it into our mold. PCR is a non-starter if the backbone is different, but peptide nucleic acids, achiral scaffolds that bind by base complementarity, can fish out templates regardless of sugar hand. You design short PNA probes against plausible motifs, purine-pyrimidine repeats, hairpins that stabilize replication, and see what sticks. Alternatively, skip letters entirely and chase translation surrogates. Shotgun proteomics, done with search spaces that include both L and D residue possibilities, can detect periodic masses and fragmentation ladders consistent with peptides built from the other side. If spectra fit only when you invert every chiral center, you've got more than noise. Here's a micro story from a pilot run that tightened our plan. A team at a salt flat set up twin bioreactors with racemic lactate labeled on one hand per tank. The gas sensors showed CO2 rising in both, but only one reactor lost a labeled hand. The other chewed the mirror. They swapped labels, repeated the run, and the preference swapped with it. Then they tried protease spikes. Our proteases erased activity in the tank, preferring L-lactate, but barely dented the other. The signal wasn't fireworks, but it didn't flinch when they poked it. That experiment didn't prove mirror life, but it proved the protocol could corner it if it was there. Antibiotics add a clean stress test. Many target stereospecific machineries, ribosomes, cell wall enzymes. You dose parallel cultures with an antibiotic and its enantiomer. If one is inert and the flipped version bites, you've mapped the handedness of a living target without sequencing a base. The same logic works with phages and viruses built for us. If nothing happens in either direction, keep that in the maybe pile, but don't overread silence. Meanwhile, build failure into the design. Spike controls with known inhibitors to show how a flatline looks when chemistry, not chirality, is the culprit. Run abiotic reactors with the same media and minerals to learn how non-living chiral biases drift over time. Every path that could fake a signal gets a test lane. Then pre-register the entire playbook, share raw data, and invite people who don't like you to try to break it. This is how you net a shadow without catching your own reflection. No single assay screams mirror life, but a stack of consistent preferences, eating one hand, resisting our proteases, flipping drug responses, leaving chiral fingerprints across pathways, builds a case the headlines Hideouts. Only where then mirror life would survive us. Everything was about to change when we asked a blunt question. If a mirror lineage can't eat our world, where could it quietly build its own? You don't hide in the buffet you can't taste. You hide where competition is thin, gradients are steady, and chemistry nudges choices. Start with brines that bite. Hypersaline pools turn our enzymes sluggish. Water activity drops, proteins misbehave. Yet haloarchaea dance there, proving life can hack extremes. For a mirror lineage, saturated salts are more than a test. They're a moat. If your metabolism evolved to prefer D proteins and those sugars, you'd want a place where our grazers and phages barely function. Enclosed basins, salinity stacks by season. The chemistry's predictable. A mirror web could idle there, metabolizing slowly, leaving thin films that standard stains ignore because the dyes bind our stereochemistry better than theirs. Slide down the pH scale to pools that eat boots. Hyperacid springs and fumaroles shave away vulnerable bonds. Our decomposers lose their edge. Acid strips membranes, ruins proton gradients, unless your toolkit is tuned to those slopes. Mineral surfaces at vents can bias absorption chirally. If calcite faces or clays sort enantiomers even a little, local microenvironments might feed a mirror preference just enough to bootstrap. Picture a biofilm thinner than paint, clinging to a flowstone where dissolved iron flips redox back and forth like a metronome. Lab assays would read trace organics, odd enantiomeric excess, likely abiotic. Then you swap to racemic probes and the pattern sharpens. Meanwhile, under our feet, kilometers of rock hold time like a freezer. The deep subsurface runs on crumbs, hydrogen from water rock reactions, sulfate trickles, slow methane. Metabolism there is patient. Generations stretch. Isolation lasts geological chapters. That's perfect for a lineage that doesn't mix with ours. You're not competing with fast surface cycles, and racemization clocks tick too slowly to erase homochirality in living systems. Pore networks and fracture waters could shelter mirror cells from predatory webs. Every sample comes up warm with abiotic signals and sparse biomass. Our primers find nothing, our cultures starve, and we shrug. But give that water a 50-50 buffet in a month, and you might see one hand fade. Hydrothermal chimneys are the other big bet. Alkaline vents lace microscopic compartments through mineral walls. Natural proton gradients hum. Metal sulfides catalyze reactions relevant to both origin stories and sustained life. Crucially, mineral chirality and flow geometry concede tiny biases. If two protometabolisms started in adjacent microcaverns, one could drift left, one right, never mixing beyond small molecules. Think of powerhouse stacks, billions of reaction cells, where selection pressures diverge on millimeter scales. From our perspective, vent scrapings look like complex organics smeared across iron and nickel. From theirs, it's home. Then there's ice, the subglacial brines threaded through Antarctica's buried lakes and Greenland's base. Salt depresses freezing points, water films persist, and nutrients arrive as whispers. Low temperature slows everything, including enzyme decay and diffusion. If mirror proteins resist our proteases, they'd be doubly stable in cold. A mirror cell could cycle once a year and still hold territory. Our probes, tuned to our membranes and markers, bounce. But circular spectropolarimetry over a meltout might catch bulk homochirality where none should sit without biology. Desert varnish writes messages we can't always read. Those glossy manganese-rich skins on rocks accrete over centuries. Microbes contribute, dust helps, chemistry does the rest. Some varnish layers show chiral excess in tightly bound organics that doesn't match local plant inputs. We chalk it up to sorting on mineral faces or photolysis quirks. What if a fraction of that sheen is a mirror film, living so slowly we mistake it for weathering? Scrape, dissolve, run chirochromatography, then run it again after an incubation on racemic feeds. If one hand thins in the scrape that sat with mirror-friendly nutrients, you're onto something. Planetary analogs help pick targets. Mars hosts chloride deposits and recurring brine activity. Europa's ocean cycles salts under radiation that can drive unusual chemistry. We're not leaping to aliens. We're rehearsing Earth's side. The logic is this: environments that select for homochirality by physical means, sorting on minerals, biased photochemistry, constrained diffusion, give mirror life scaffolds to stand on. Identify those scaffolds here, test protocols, then carry that playbook to places where our biosphere never booted. Here's a micro story that stuck with a field team. A researcher in Chile's high-altitude salars swabbed a purple slick above an evaporate layer. In camp, their standard enzyme panels did nothing, a racemic lactate. Microcosm, thrown together as a why not, crept upward in CO2 after a week at night temperatures. They chased it with Chiral LC. The D-lactate pool thinned, the L pool barely budged. They repeated, flipped labels, got the mirror result. Then they added our proteases. Activity in one set collapsed. The purple slick set barely flinched. They didn't call a press conference. They called for more salt, more time, more controls. One more hideout sits in plain sight. Human systems. Water treatment plants, industrial fermenters, hospital plumbing. Biofilms accumulate where chemistry fluctuates and cleaning selects for outliers. If our disinfectants and enzymes bias against our own handedness, the mirror tail of a mixed population, if it exists, could become the head in those corners. A drain that resists every usual biocide but falters, when you swap to enantiomeric detergents, would be less a nuisance and more a signal. It's a place to watch, not fear, unless we ignore it. So we choose hideouts where selection lowers the noise, where our biology moves slowly, where chirality can lean without toppling. If a mirror lineage survives us, this is where it waits. And if we're careful, this is where we'll meet it first. The Firewall. Why mirror life doesn't mix with ours. While those hideouts simmered away from our attention, something else explained why we hadn't already collided head-on. Geometry builds a firewall. Not a wall you can touch, one made of shapes that refuse to handshake. Our enzymes were carved by evolution to clasp L-amino acids and D-sugars. Flip the hand and most interactions slip. Metabolism turns into a room full of doorknobs that don't fit any key you have. Start with breakfast-level biology. Transporters in your gut pull specific sugars across the lining, but they're asymmetric pockets. Glucose tastes sweet to your tongue, but ghost walks past those pumps and exits the other side because nothing grabs it. Scale that to a cell trying to eat a mirror bacterium. The praised proteins and polysaccharides present surfaces with the wrong twist. Our digestive enzymes bite air. Even scavenger enzymes that chew debris stall because their catalytic centers are stereospecific. That's nutritional isolation, the cleanest reason parallel food webs could sit centimeters apart and never meet. Drugs don't cross the gap either. Most frontline antibiotics hit ribosomal RNA folds, topoisomerases, cell wall assembly lines, targets with chiral architecture. Switch to a ribosome built from mirror components, and those binding sites invert. The drug's shape no longer complements the pocket. Activity collapses. It's not universal. Some antibiotics disrupt membranes or redox broadly, but the heavy hitters are shape readers. In a mirror lineage, you'd expect our staples to miss, and in some cases, their enantiomers to gain potency. That's a quick, diagnostic, and a sobering clinical blind spot. Ecology then splits like a forked river. Decomposers shape soils by cracking plant litter and recycling nitrogen, phosphorus, and carbon. If a mirror decomposer community rises in a patch, it can't easily eat our detritus. Our side can't eat theirs. You get parallel lanes of nutrient flow with limited bridges at small molecules, gases, simple acids, minerals. Carbon dioxide mixes fine. Acetate can be made in either world. But the big traffic, proteins, polysaccharides, nucleic acids, stays on its lane. That's why a mirror web could persist without instantly starving or transforming ours out. It sips what it can, synthesize, and what diffuses across as simple metabolites. There's a caveat with time. Amino acids racemize slowly in dead matter, L drifting toward D, so sediments creep toward balance over centuries to millennia. that muddies chemical signals but doesn't feed living metabolisms. Racemization is too slow and random for cells to rely on. It also means a mirror carcass won't become lunch for our microbes anytime soon, because the chiral centers don't flip on useful timescales. Persistence rises, mixing stays low. Immunity plays a double game at this border. Pattern recognition receptors bind motifs like lipopolysaccharides and peptidoglycans with set stereochemistry. A perfect mirror could slide past some sensors, a kind of molecular stealth, while half-matches could misfire and spark exaggerated inflammation. That's the mirror septic shock scenario clinicians worry about. Fragments that don't fit normal tolerance profiles, tipping alarms the wrong way. It's theory until we test it, but it follows from how tightly innate immunity reads twist and spacing. Here's a micro story from a containment drill that drove the point home. A lab flagged an odd contaminant in a bioreactor that processed agricultural waste. Routine plating showed nothing. ATP tests were low, yet gas production ticked along and the viscosity rose. Classic biofilm behavior. They tried standard proteases, no change. They switched the feed to a racemic amino acid mix with labeled the residues and watched the label vanish while L sat untouched. When they dosed ampicillin, nothing happened. Then they dosed mirror-configured ampicillin they'd kept for controls. The film thinned. The contaminant hadn't been eating their inputs. It had been living off small acids it made itself and building structures our enzymes couldn't dent. Starved on our buffet, it thrived on a mirrored snack bar. The firewall had teeth. Now this separation is safety, until it isn't. Interfaces form where small molecules flow, like root zones, wastewater gradients, or medical devices. If a mirror population blooms on a catheter and our antibiotics can't touch it, minor infections become stubborn biofouling. If mirror biofilms establish in food processing lines, routine CIP cycles tuned for our stereochemistry might polish the wrong side. Industrial fermentations could stall when trace mirror metabolites bind inhibitors that our sensors don't flag. None of this is doomsday. Growth rates, energy limits, and environmental stress cap outbreaks. But each interface is a weak point where parallel worlds brush sleeves. Flip the perspective, and the firewall gives us a lever for containment. If mirror cells can't eat our world, the open environment is a starvation buffer. Feedstocks become locks, cut mirrored nutrients, and any escapees run out of calories. Our proteases ignore their proteins, which is bad for cleanup, but good for confinement if we stock facilities with mirror-only food and chiral tripwires. Genetic safeguards can stack atop geometry, design pathways that require mirror cofactors never found outside. Even simple heat or UV remains universal. Photons don't care about handedness, so physical kill switches still work. We shouldn't romanticize the barrier. It's strong, but not absolute. Mixed chemistries can couple through shared gases, and evolution is opportunistic, yet the main story holds. Two biospheres can pass like trains in fog, close enough to feel the wind, far enough to miss each other's stations. That distance explains the quiet, and warns us where to look when silence breaks. The end of biology scenario, plausible paths to disruption. Everything was about to change when someone asked a rude question out loud. What if the firewall cracks where our systems are thinnest? Not apocalypse, just cascading annoyances that add up to a very bad year. Start in the dirt, because agriculture lives or dies there. Soils run on decomposers that unlock nitrogen and carbon from plant litter. If mirror decomposers establish patches, they won't digest our fallen leaves or crop residues. Their enzymes slide off the polymers. At first, you just see slower turnover, cover crops lingering, residues stubborn under the plow. But nutrient cycling is a relay race. If one leg stalls, the next runner, nitrifiers, denitrifiers, mycorrhizae, waits with nothing to pass. Farmers would dial up fertilizers to compensate. Yields might hold for a season, then costs jump, then micronutrient imbalances creep in. In parallel, mirror biofilms on root surfaces could siphon small acids and gases without feeding the plant. The field looks fine, the soil test reads off. Agronomists swap varieties, tweak irrigation, and complain about weird seasons. Underneath, chiral pools drift. Amino acids in residual matter show rising diffractions where L should dominate. The tractor keeps moving while the chemistry slides sideways. Meanwhile, inside tanks and stainless steel cathedrals, food processing hits snags nobody trained for. Fermentations are choreography, yeast on sugars, bacteria on lactose, enzymes cutting proteins on a schedule. If mirror contaminants seed a line, the standard starters can't eat their substrates, and the contaminants can't eat ours. Both sides waste time. Yogurt stalls at half set. Sourdough starters turn listless. Industrial proteases ignore mirror-stabilized films that build on heat exchangers, and CIP cycles polish the wrong side. QA screens, tuned to our enzyme panels, say minor deviation. Then a plant loses a week chasing phantom inhibitors. The fix, when it arrives, is disturbing in its simplicity. Swap in the enantiomeric versions of a few critical enzymes and see the fouling drop. Maintenance logs become chirality diaries. Hospitals feel it sharper. A stubborn wound refuses to debride despite broad-spectrum antibiotics and enzyme dressings. Cultures come back no growth, PCR says absent, but the biofilm looks busy. Surgeons flush, resect, and start over. Sepsis screens, built to catch classic endotoxin profiles, misfire, either too quiet or too loud. One ER writes up a streak of culture-negative fevers that spike hard after debridement. Fragments in the bloodlight innate sensors in patterns that don't fit training data. Pharmacies scramble when ribosome-targeting drugs underperform, and infectious disease teams trial mirrored enantiomers pulled from teaching collections. In 3 cases, activity flips like a chirp. Drug B fails, drug B mirror bites. That's not a cure, it's a map. Procurement chains, not used to ordering D-configured peptides or mirror beta-lactams, jam. Shelf lives improve. D proteins resist our proteases, but dosing and toxicity data are thin. Clinicians become field biochemists in white coats. Biofouling scales up beyond hospitals. Municipal pipes sprout films that shrug off enzymes and surfactants, backing up neighborhoods. Desalination membranes foul in patterns plant managers haven't seen, with mirror EPS matrices that resist breakdown. Wastewater plants watch flock behavior change. Aeration energy ticks up. Operators add biocides and see diminishing returns until someone tests the chiral twin of a detergent and the sludge relaxes. Insurance actuaries, not biologists, are first to notice the trend line. Then planetary protection walks in, because the line between Earth and elsewhere is thinner than it used to be. Sample return capsules bring sealed rocks from Mars or an asteroid. Clean rooms hum. Agnostic mass spectrometry sees a bias in enantiomeric excess that doesn't match our baselines. The lab goes slow. Controls, blanks, chiral LC. If it's biological and mirrored, there's a non-zero chance it can persist in a corner of our environment where our enzymes never reach. Dry, cold, salted, or flip it. A mirror lineage incubated on Earth, contained well for years, rides a film on a valve seat to a Mars simulator. Forward contamination with a different handedness wouldn't infect a red planet's hypothetical life. It would graffiti the chemistry, complicating every future detection. In either direction, tiny leaks create decades of interpretive fog. Here's the gut punch. None of this collapses society. Growth rates for mirror microbes would be constrained by the same physics ours obey. Energy limits, diffusion, temperature. They can't eat our biomass, we can't eat theirs. Interfaces are narrow, but the friction at those seams is expensive. A harvest season shifts late when decomposition lags. A cheese plant idles. A hospital revises protocols and burns through its budget on custom drugs. Multiply inconvenience across continents, and you get supply chain wobble. Constraints help us breathe. Racemization is slow. It doesn't hand mirror life a bridge into our biochemistry. Environmental chiral baselines take work to move. You need sustained selection, not a weekend bloom. Mirror biofilms might resist our proteases, but UV light and heat ignore handedness, and oxidants don't care about geometry. Most problems respond to physics. So what turns this from bad month to bad year is denial. If we cling to our assays, our media, our go-to-fixes, we'll chase ghosts while films thicken. If we pivot, chirality-aware screens in clinical labs, enantiomeric detergents in plants, racemic feeds and environmental tests, the cascade slows. The firewall that kept us apart can be repurposed as a moat. Feedstocks become locks. Monitoring becomes early warning. The end of biology isn't literal. It's the end of biology as a monoculture of one hand. The sooner we accept that, the steadier our grip on the wheel. The upside, medicines, materials, and clean tech. Everything we just walked through sounds like headache and hazard, but the same firewall that complicates life outside gives us leverage inside. If we keep mirror chemistry in a box, it becomes a toolkit our biology can't easily spoil and pathogens can't quickly outsmart. The trick is discipline, thick walls, smart locks, and problems worth solving. Start with medicine. Antibiotics fail when targets evolve or when biofilms shield them. Mirror antibiotics step around both. A D-peptide antibiotic, built from D-amino acids, slides past our proteases, so it doesn't get chewed up in serum or the gut. It also dodges many bacterial enzymes that evolve to neutralize L-handed drugs. Here's where it gets useful. Bind a universally conserved pocket by shape, not stereochemistry, and most pathogens won't have seen that geometry under selection. Resistance can still evolve, but the starting line moves. And because our bodies don't make the proteases in bulk, dosing windows widen, longer half-life, fewer shots. In trials, you'd pair every D drug with non-chiral kill switches, UV, heat, to keep containment honest. Now flip to antivirals and vaccines. Peptide epitopes made from D residues don't get clipped by L proteases, so they can sit on a shelf for months without cold chain drama. You can stabilize mimics of viral fusion loops in D form, using them as decoys that absorb binding without triggering off-target immune cascades. Adjuvants become more predictable when the active bits don't degrade while you're drawing syringes. Hospitals that struggle with storage can hold stock longer with less waste, especially in disaster zones where refrigeration is a rumor. Nutrition gets a surprising gift: sweet without sugar. L-glucose is the classic example for us. Tastes sweet, passes through. Mirror. Sugars from a mirror bioreactor push that further. You can build complex mirror oligosaccharides that hit sweetness receptors, but refuse digestion, turning into low-calorie bulking agents that don't feed gut microbes the way regular fibers do. That's a double edge. In a clinic, you can craft diets for metabolic syndromes that deliver mouthfeel and satiety with minimal glycemic load. In the wider world, you don't want to flood sewers with substrates our ecosystems can't process. Containment keeps the pantry clean. Enzymes are the workhorses of clean tech, and mirror versions come with quirks that help. D proteins shrug at many proteases, resist thermal denaturation a bit longer when designed right, and hold shape in solvents that would tease L proteins apart. Imagine a remediation skid at a refinery leak site. You spike a closed loop with mirror dehalogenases tuned to crack stubborn chlorinated solvents. They hum for weeks without fouling, and when the job's done, the enzymes don't persist outside because the environment offers no calories their microbes can use. That's targeted cleanup with a built-in ecological off-switch. Material science loves stability, and D proteins are patient. You can build mirror collagen analogs that keep tensile strength in heat and humidity cycles that trash the L versions. Medical implants, coatings on stents, scaffolds for tissue growth could use mirror matrices that resist enzymatic breakdown until you ask them to go, using non-chiral triggers like light or temperature. Data storage edges in too. Protein-based archives need decades of integrity. Mirror polymers buy time against the slow creep of biology and rogue proteases. Stored in dry, cold vaults, they approach that immortal behavior engineers crave, not forever, but far longer than our current media. Then there's biocontrol. We fight invasive species and crop pathogens with chemistry that bleeds into waterways. Mirror enzymes let you go narrow. A D-cutinase sprayed in a controlled greenhouse breaks down a mirror polymer mulch at end of season without touching plant cuticles or soil proteins. In a mine runoff channel, a mirror sulfidase chews target compounds without crosstalk in a downstream wetland. The key is always the box. Run it in line systems, monitor effluent with chiral tracers, and treat waste streams until the signal is gone. We also get a diagnostic edge. Hospitals and plants can install paired assays, L&D, at choke points. If an unexplained fouling pops, you flip a switch and test both hands. That shortens the mystery phase. You can even build mirror decoys into surfaces, coatings that bind mirror EPS preferentially, pulling early biofilms onto sacrificial patches you can UV blast or heat pulse without dosing whole systems. Ethics keeps the rails straight. None of this leaves the lab without layered lock slab. Feedstocks inside are mirror only. If a pump leaks, the outside world is starvation. Facilities run negative pressure, HEPA, and chiral filtration on waste. Genetic dependencies hardwire mirror cofactors that don't exist beyond the walls. Auditors sample effluent with circular dichroism and chiral LC, posting data in public dashboards. And before any pilot, communities near plants see the plan, the failure modes, and the shutoff valves. We learn from the Arsenic saga. Oversell nothing, pre-register everything, invite rivals to watch. Here's a micro story that shows the promise without romance. A burn unit tested a dipeptide hydrogel as a temporary barrier. It stayed intact through daily rinses, blocked colonization by common pathogens, and peeled away clean on schedule with a brief UV exposure. Nurses liked it because the dressing changes were calmer. Pharmacists liked it because stock didn't expire every other week. The team didn't call it a revolution. They filed the data and extended the trial, one ward at a time. That's the rhythm. Mirror chemistry isn't a silver bullet, it's a different screwdriver. In a box, with rules, it lets us solve problems our enzymes keep undoing. Outside the box, it's friction. The wall between those worlds decides whether we get medicines that last, materials that hold, and cleanup that finishes. Containment, or a long list of preventable headaches. How you keep mirror life in a box. To use the upside without inviting the headaches, you build walls, thick ones. Then assume they'll be tested. Start with the room itself. Biosafety upgrades aren't decorative, they're physics. Negative pressure keeps air flowing inward, not out. 2 doors between lab and hallway mean you always step through a small wind. HEPA stacks catch particles, but we add a twist. Chiral filtration on liquids. Waste lines run through cartridges packed with chiral resins that bind one enantiomer set harder than the other, so any mirror polymers or metabolites stall for collection. Autoclaves handle what heat can reach. Oxidants follow for what geometry resists. Inside, the pantry becomes a lock. Mirror-only feedstocks make the outside world the desert. Media are racemic only for tests. Production runs feed strictly mirrored sugars and diamino acids, dyed with distinctive stable isotopes, so you can sniff leaks by mass spectrometry in parts per trillion. Every container wears a tracer signature. Think of it as a chemical watermark. If a droplet escapes, the sump knows before the shift ends. Starvation is the first containment layer, surveillance is the second. Then you wire in genetic dependence. Metabolic pathways are edited, so core enzymes require mirror-specific cofactors, synthetic deflavins or mirrored p-tyrines that don't exist outside the building. Pull the feed and the pathways stall. On top of that, add oxotrophies for non-canonical mirror amino acids the facility alone can supply. The organisms can't scavenge from our environment. The chemistry simply doesn't fit. It's a diet that doubles as a dead man's switch. Kill switches sit on the same shelf as fire extinguishers. Visible, tested, boring. UV floods ceiling to bench. Photons don't care about handedness, so a 2-minute blast cross-links nucleic acids of any stripe. Thermal tripwires flank drains. If a sensor flags pressure where it shouldn't be, inline heaters spike to pasteurization. Chemical scrubs sit downstream. Peroxide and parasitic acid baths tuned to oxidize stubborn mirror EPS matrices our proteases can't touch. You don't trust one hammer, you hang a rack. Waste management gets its own choreography. Effluent is split into streams, solids to surveillance, watching the planet for a second biosphere. While the containment crew runs drills inside, the rest of us need eyes outside, quiet, persistent, and chirality aware. Surveillance starts with a map, not a hunch. Environmental baselines of enantiomeric excess across soils, waters, aerosols. You grid the planet like a weather service. Field teams pull cores, dip bottles, and collect dust on rooftops. In the lab, chiral chromatography splits twins, and isotope ratio mass spectrometry checks whether one hand is drifting faster than chemistry alone can explain. Most places will read boring, near racemic for many free molecules, strong Lessnitures in biomass-derived pools. That lets anomalies stand out instead of getting lost in averages. Spectropolarimetry layers on a global lens. Circular polarization signals, weak but telling, betray bulk homochirality when reflected off dense microbial mats and leaf canopies. We already use it to read photosynthetic fingerprints. Now we tune it to flag places where chiral magnitude is high, but the sign doesn't match expected plant cover. A wetland that looks normal to RGB might show a polarization tilt nobody logged before. You don't send a press release. You send a cooler and a racemic feed kit. Water is the fastest gossip, so rivers and treatment plants become sentinels. Install paired enzyme assays, L&D, at influent and effluent, and effluent. If an unexplained inhibition pops on the L panel while the D panel hums, that's not proof, it's a breadcrumb. Add inline chiral LC on dissolved organic carbon to watch seasonal swings. After a storm, urban runoff spikes amino acids and sugars. The ratio of hands should track plant litter. If it tilts the wrong way in a recurring pattern, someone upstream is hosting unusual chemistry, industrial, mineral, or biological. You trace it like a leak. Origins, how mirror life could have started. While the surveillance map comes online, a tougher question hums underneath. How would a mirror lineage start at all? You don't get homochirality for free. Chemistry, left to itself, mixes hands 50-50. Something has to tip the table, then lock the tilt in. Start with parallel genesis. Early Earth wasn't one beaker. It was a patchwork of wet-dry cycles, tidal flats, hydrothermal pores, and frothy shorelines. It's not hard to sketch two neighboring labs nature could run at once. On a sunlit shoreline, thin films of organics bake and soak, concentrating sugars and nucleotides. In mineral pores a few meters away, alkaline fluids seep through iron-sulfur walls, feeding reductive chemistry. Each site has its own rhythms, UV light and evaporation over here, steady gradients and metal catalysts over there. If chiral, selection sneaks in differently at each. You can imagine one protosystem committing to these sugars and L-amino acids, the other to their mirrors. After that, feedback keeps them apart. Autocatalytic loops favor their own kind. Mixed chains stall. Crosstalk feels like sand and gears. Minerals help set the bias. Louis Pasteur learned chirality's reality by hand space. Mirror life beyond Earth and back again. 3 days earlier, in a clean room lit like a midnight pharmacy, a tech watched a needle on a mass spectrometer lean a few parts per thousand toward the wrong hand. The rock was sealed, the instrument scrubbed, the blanks flat. It was just a hint, an enantiomeric excess where none should be. They didn't cheer. They started logging. To understand why that tiny tilt matters, look outward. Mars holds chloride plains, recurring brines, and perchlorate spiked soils that stress our enzymes, yet might shelter slow metabolisms. Europa and Enceladus run dark oceans under ice, laced with salts and powered by tidal kneading. Radiation sculpts chemistry at their surfaces. Minerals sort molecules by subtle fits. If chirality can be nudged by light, crystals, or flow, then off-world settings could pick a different hand than Earth did, and keep it. Space agencies have started shifting from, is there our biology out there, to is there any consistent biology out there? That's why instruments now aim for agnostic signals. On orbiters and landers, spectropolarimetry looks for circular polarization patterns that scream bulk homochirality in reflected light. It won't tell you left or right, it tells you consistent. Drill cores feed micro-GCs and chiral LC into time-of-flight mass spec, splitting twins before weighing them. A racemic world averages out. A living one leans. Now rewind to the lab because sample return raises the stakes. When a capsule carrying Mars regolith or plume ice clicks into a glove box, the detection pipeline can't be parochial. You run parallel paths. One assumes Terran backbones, PCR on D-sugar scaffolds, L-amino acid proteomics. The other refuses assumptions, peptide nucleic acid probes that bind by base complementarity regardless of sugar, and LCMS libraries that allow D-residue ladders and flip fragmentation rules. You pair those with chiral separations and stable isotope spikes to see who eats what when warmed, moistened, or fed. Then you spend days trying to prove the signal is your fault. Forward contamination ethics tug the other way. If we carry mirror chemistry to Mars, it won't infect hypothetical locals by eating their lunch. It'll graffiti the chemistry and confuse every future test. A D-protein biofilm on a drill bit leaves residues that our Earth-leaning assays might mistake for natural bias. So hardware now lives under planetary protection regimes that treat handedness as part of bioburden. Tools see dry heat sterilization, peroxide plasma, UV floods. Witness plates get both L and D assays. Clean rooms map enantiomeric excess of swabs like weather maps. If a corner drifts, they tear it down and rebuild. Here's where the moon earns a new job. Lunar labs give us gravity, vacuum, and a world with no biosphere to confuse. You can run mirror biochemistry in hardened modules with closed loops and radiation you can't ignore. It's a disciplined platform. Racemic feedstock, ship sealed, waste cracked under UV and heat, effluents vitrified. If a mirror lineage ever needs scale for medical manufacturing or materials work, you put the pilot there. The outside is absolute starvation. If a seal fails, dust gets in, nothing living gets out. Back on Earth, the mirror septic shock worry informs protocols for handling unknowns. Clinicians watching culture-negative fevers here have taught spacecraft folks a lesson. Prepare for fragments that ring the wrong immune bells. In a sample return workflow, you don't just test for growth, you test for interaction. Small aliquots meet human cell lines behind extra glass. You watch cytokines, not colony counts. If inert fragments spark storms, you add layers of blunting chemistry before anyone opens a second glove. Unknown to many, deep in the instrument stacks, a quiet revolution helps arbitrate claims. Tandem mass spec trained on both worlds at once. Machine learning models, fed with synthetic D and L libraries, score spectra without assuming our side first. That matters when you get a hit that only fits if every chiral center flips. A human can miss that. A well-trained classifier flags it in milliseconds and asks for orthogonal confirmation. NMR coupling constants that match mirror geometry. Circular dichroism with an unexpected sign. There's also a politics to all this. The Arsenic saga taught teams to slow their roll. Agencies now pre-register detection trees. If signal X appears, we run paths A, B, C. If not, we pause. Datasets drop publicly with raw files and blank runs. Independent labs mirror the analyses, literally and figuratively. A mirror. Claim, if it comes, we'll crawl through committees before it reaches a podium. That's not bureaucracy, it's memory. Here's a micro story from a plume simulator that pulled theory into fingers. Engineers injected racemic amino acids into a cryogenic jet, let ice grains form, then warmed them through a vent mock-up. Chiral LC on the melt saw a tiny lean that matched no mineral template they'd included. They suspected contamination. They rebuilt the rig, replaced seals, scrubbed lines, and the lean stayed. Then they introduced a D protease as a tracer enzyme in a sealed side channel. Downstream, the D pool fell faster. The L pool barely moved. It wasn't proof of life, it was proof their pipeline could catch preference under punishment. If mirror life exists off-world, bringing it home isn't the prize. The prize is clarity. A second data point for what life means, handedness and all. The cost of haste is decades of doubt. So we build instruments that don't flinch, labs that don't leak, and stories that wait until the boring graph refuses to go away. On another world, then back again. The case files, clues, red herrings, and what would count as proof. While that plume simulator kept humming, teams started assembling case files, hits, hints, and the traps that fool smart people. First rule: separate biology's signature from geology's habits. Minerals can twist chemistry. Calcite, clays, even ice can sort enantiomers a little. Sunlight can bias reactions through circularly polarized light in the sky. Those routes leave patterns, but they're shallow. Abiotic chiral excess usually fades with distance, dilutes with mixing, and rarely repeats the same bias across unrelated molecules. Biology, by contrast, is stubborn and coordinated. It picks a side once, then stamps that choice on amino acids, sugars, lipids, and polymers together. So the red herrings go first. A varnish scrap with a 2% D excess on one amino acid could be mineral templating. A lake with L-leaning sugars near dense algae? That's us. A meteorite with non-zero enantiomeric excess from space chemistry? Interesting, but not alive. The bar for life starts where coordination shows up. If proteins, polysaccharides, and nucleic acid surrogates all display consistent handedness, and the same hand shows up in metabolic flux, that's when eyebrows rise for the right reasons. Multi-omics makes that coordination visible without forcing Terran assumptions. You run proteomics with search spaces that allow full D residue sequences alongside L if spectra only fit when every residue flips and the fragmentation ladders line up across peptides from different samples. That's a pattern, not a glitch. Metabolomics then scans small molecules with chiral LC up front, mapping which enantiomers exist and how they respond to perturbations. Lipidomics watches backbones and head groups, glycerol chirality, sugar attachments, to see if the whole toolkit mirrors ours rather than cherry-picking a few stereocenters. Meanwhile, NMR confirms the 3D geometry independently, and circular dichroism tallies bulk homochirality. None of these alone is proof. Together they close exits. But molecules don't just sit. They eat, build, and divide. Growth tests pin that down. You set up paired microcosms with racemic feeds and stable isotopes in just one hand. Respiration upticks, cell counts by agnostic stains rise, and the labeled pool thins, only when the label matches the appetite. Flip the label, watch the preference flip. Layer and protease challenges. Our L proteases erase activity for L-built systems. They barely dent D-built ones. Then run the antibiotic mirror test. Dose with a ribosome-targeting drug, and it's enantiomer. If one fails and the mirror works, you've mapped stereochemistry of a living target without a single PCR cycle. Cross-incompatibility seals the biological case while guarding against artifacts. Phages and viruses adapted to our biochemistry should ignore a mirror host. If you spike cultures with our pathogens and see nothing, no lysis, no integration, no cytopathic effect, then try mirrored substitutes in the same setup and see activity return. The story coheres. You're not proving immunity, you're showing orthogonal machinery that refuses our keys but turns for its own. All of this has to ride on clean rails. The Arsenic episode burned one lesson into muscle memory. Controls are not decorations. Every feed gets paired blanks that track carryover. Every separation includes racemic standards to catch column drift. Isotope spikes quantify loss with math that doesn't care about enthusiasm. Protocols go up pre-registered, so nobody can tuck a new step in after peeking at results. And the data don't live behind paywalls. Raw files drop with the claim, so rivals can re-index spectra with their own tools and catch your blind spots. Then comes replication, not in spirit but in plumbing. Independent labs receive sealed samples, run the same racemic feeds, the same DL assays, and get the same tilts within error. They swap out instruments, Orbitrap for TOF, NMR field strength up or down, and the geometry holds. They move the whole show to a different environment. Salt flats 1 month, subglacial brine the next. If the signal follows the microbe, not the mineral, and the toolkit reads the same every time, the claim graduates from interesting to robust. There are still sirens to ignore. A single, strong chiral signal in a hostile setting can tempt headlines. Resist it. A neat D protein that resists our proteases could be a synthetic contaminant. A mirror-leaning metabolite suite might ride in from industry. That's why provenance and chain of custody read like detective novels. That's why environmental baselines sit on servers before the hunt begins. So anomalies stand in context rather than legend. Here's the sniff test. Teams use before calling anyone. Does the candidate system display coordinated homochirality across major biopolymers? Does it grow on one hand of racemic substrates while ignoring the other, with isotope flux to prove it? Does it resist our stereospecific enzymes and drugs, yet respond when those tools are mirrored? Does heredity show up with achiral binders or translation surrogates? And can 3 labs using pre-registered steps reproduce those answers on fresh samples? If you can say yes down that list, you don't have a red herring. You have a living mirror. The meaning and the ask. If mirror life exists, we won't tweak biology. We'll meet a second engine that solves life's problem set from the other side, forcing us to rebuild definitions and tools. Want the next chapter in this hunt? Subscribe, share this with a friend who labels their sourdough jars, and watch our follow-up on prions to see how life bends its own rules. Then, tell us where you'd search first. Your maps might lead the next expedition.